Good morning, everyone. Today we are delighted to have Professor George Davey Smith with us at CBR. Professor George Davey Smith is a distinguished clinical epidemiologist who has served as a professor of clinical epidemiology at University of Bristol since 1994, honorary professor of public health at the University of Glasgow since 1996, and visiting professor at the London School of Hygiene and Tropical Medicine since 1999. He has been the scientific director of A1 longitudinal study of parents and children and editor-in-chief of the International Journal of Epidemiology. He is a highly cited researcher. He is also a fellow of the Royal Society of Edinburgh and the Academy of Medical Sciences UK, and was elected as a foreign member of the Royal Netherlands Academy of Arts and Science in 2019. He will be delivering a talk on Mendelian randomization, what it was, what it is, and what it should become. We are truly honored to have him with us today. Over to you, Professor George.
Should start wearing a collar. It's okay. Perfect. Okay. Great. Thanks. Yeah. Thanks very much. Yeah. So I'm going to talk about Mendelian randomization, and I'm very happy to be. If you've got questions while I'm going through, just ask me. You can interrupt me and ask me them, and then there'll be plenty of time afterwards to discuss things. So, and I mumble. I know that I mumble, and if you don't understand something because I'm mumbling, then which is very likely, then just ask me to say it again, because I understand I mumble and I have an accent and etc. So, so I'm going to talk about a method called Mendelian randomization, first introduced 20-odd years ago. And the method was introduced at a time when the failure of conventional observational epidemiological studies to get the right answer about important questions was becoming increasingly evident, because there were some very high-profile randomized controlled trials that came out testing hypotheses that were the best hypotheses available from observational epidemiology for things that could be modified to reduce cardiovascular disease and cancer in particular. And at the cost of billions and billions of dollars, these randomized trials spectacularly failed. These were seen, many of them were in the area of nutrition, in particular micronutrient supplementation, where observational studies suggested that vitamin E supplements, one of the first spectacular failures, would reduce coronary heart disease risk by 35%. This was so well publicized in the US that up to nearly 50% of the population at one point in time started taking vitamin E supplements. And then when the randomized trials were done, massive trials were done with up to 13 years follow-up, nothing. Absolutely no effect. And this was the best candidate. Precisely the same story was seen for vitamin D, vitamin C, folate, for selenium that was supposed to protect against prostate cancer, for homocysteine, and many other things, beta-carotene. The list is a very long list. That's why the trials cost billions of dollars. And also drugs that were targeted at lowering biomarkers, things that were thought to be causal. Many of these happened. Probably the most publicized one was HDL cholesterol, high-density lipoprotein cholesterol, which was known as good cholesterol. I mean, in popular discourse, it was called, this was the good cholesterol that was supposed to reduce coronary heart disease risk, and drug trials that raised HDL cholesterol had absolutely no effect on disease outcome. So the option was either to carry on as business as usual, which was failing rather dramatically, or to think of other approaches. So the reason why these trials, the reason why trials were done on factors that were actually not causal was because in observational studies, you measure some factor like HDL cholesterol or like vitamin levels, antioxidant vitamin levels in the blood, or you measure behavior, like dietary behavior, etc. You follow people up, you see if those factors predict events, heart attacks, cancer, whatever your interest is. And of course, everyone realizes that there is confounding, where a factor which causes the outcome, like smoking, for example, might lower your level of vitamins in the blood, or might influence your diet. So there's very many confounders that could exist, where the factor that you're measuring is influenced by these confounders. And probably the most insidious form of confounding is reverse causation. And that is when the early stages of the disease process influence your ostensible exposure. And this can happen decades before you actually manifest disease outcomes. The disease process is developing. So think of a disease process like atherosclerosis. As the name implies, atherosclerosis is an inflammatory condition. It is the incorporation of cholesterol level into the intima of your arteries, and particularly your main arteries. And then there's oxidation of the lipid, and this instigates an inflammatory process. So that is a silent condition. Atherosclerosis developing is silent until you have a heart attack or an ischemic stroke, and it develops over decades. When American soldiers in the Korean War, 1956, were autopsied, the 19-year-olds, the youngest of them, already had atherosclerosis. And interestingly, when the same happened in the Vietnam, a decade later in the Vietnamese conflict, there was evidence it was decreasing a little bit. And that actually said what was then going to happen, which was coronary heart disease rates in the US started falling from the 1970s. It was presaged by saying that something was already happening to the disease process. But the reason I go on and on about this is the insidious nature of reverse causation. And that people say, oh, well, we'll only look at things we measured 5 years ago, and that will deal with reverse causation. It doesn't. If you look at the genetic scores for Alzheimer's disease, those genetic scores predict weight loss happening from about 50. People start losing weight years before they, at a group level, start losing weight years before disease develops. So that very long intro is because people just say, well, if you want to know about how modifiable causes, how they influence disease, why don't you just study the causes? Just do diet surveys, just measure people's HDL levels. That will tell you the answer. The point isn't, for someone of my generation in epidemiology, these failures were spectacular. These were the things that everyone was putting their money on. This was literally billions were put onto these things, and they failed. And I couldn't understand why people just sort of forgot about them. They went on to the next thing. They didn't try to address the question about why had they got these things wrong. So that is saying why Mendelian randomization. Why it was why something new approaches were required. because it's just impossible to exclude confounding and or reverse causation, and we know that these generate spurious findings. So the trick is incredibly simple. People complain that Mendelian randomization is difficult to understand, but it's not. The trick is remarkably simple. It's if you've got a modifiable exposure, something like cholesterol level, and it is heritable, and virtually everything is heritable to an extent. It's remarkable the extent to which heritability stretches to things which people might think aren't obviously heritable, like risk-taking behavior patterns. The one thing that isn't heritable is whether you get a head or a tail when you toss a coin. That's about that's not heritable. But most things are heritable to a degree. So the exposure has to be heritable, but then importantly, and something I will repeat again and again and again, there have to be genetic variants that relate to that exposure, enough variants that relate to that exposure relatively directly. Because and I'm going to come back to this point again, so I'll carry on, but then I'll come back later again and again about that. This is the point, that the variants, because everything is heritable, if you do a genome-wide association study, and just to say, and that, the study where you have genetic variants, now 10 million genetic variants with imputation across the genome, and you get all those 10 million variants genotyped, and something is heritable, you will find variants that relate to any trait. But some of them will relate through such a convoluted pathway that they relate to many, many other things. You need to have variants that can actually tell you something directly about the trait. Now, for something like, for many circulating biomarkers, virtually all circulating proteins, or proteins actually that don't circulate as well, there are variants. The proteins have genes that produce them, and there will be variants actually in those genes. We have less than 30,000 genes, but we have a vast amount of regulatory material which regulates and co-regulates them. But for something like cholesterol level, you have a variant in, for example, in the receptor. So you have LDL cholesterol, which is the bad cholesterol, low-density lipoprotein cholesterol, and you have the low-density lipoprotein cholesterol receptor, and genetic variants in that influence your cholesterol level that's circulating, because they influence how much of the cholesterol is taken up by liver in particular, which then reduces your circulating cholesterol. Now that's a nice story, because your modifiable exposure is low-density lipoprotein cholesterol. And I'm a very simple person, and when the name of your exposure is in the name of the gene, then it's quite plausible that is having a pretty direct influence on the exposure. So the trick is that if you have a genetic variant that relates to your exposure, then the genetic variant will not be confounded in any way by conventional confounders. Your genetic variant these are germline genetic variants, the ones you are conceived with. Now, you get somatic mutation, etc. After conception, there's relatively low levels of that to begin with, luckily, but it increases. But the variants that you genotype, when you do GWAS, genome-wide association studies, you are genotyping people's germline, the germline genetic variants that they received from their parents. That's what you genotype. You're not looking at things that have changed since conception, that have been mutated or whatever since conception. the findings that you get, and people take a lot of care to ensure this, are from what you are randomized by at conception. That's what. Now, smoking can definitely influence your cholesterol level. Weight loss can influence your cholesterol level. Atherosclerosis can influence your cholesterol level. Those things cannot influence your germline genetic variation. That is what you got 9 months before you were born. You can't reverse time's arrow and change that. There is no conventional confounding of germline genetic variation. So it's a really simple trick. The problem with observational epidemiology is confounding and with reverse causation being a particularly insidious form of confounding. If you look at the genetic variants, they cannot be confounded. Question?
George, how about epigenetics here? Is that it's not important enough?
No, it's not important enough because at a group level, epigenetics, whatever happens to that, if you group the people by the LDL receptor variant and the other, you get a group level difference. Epigenetics never compensates for that group level difference. That's quite. It's often get that question, but it doesn't. No, the epigenetic modifications don't change the group level difference. You can't. You cannot, assign causality to individual events generally. A personalized medicine is very, you often hear about the term personalized medicine. There's no true personalized medicine at the individual level. If you're going to make causal inference about something that you think affects human health, then everything has to be based upon group level group level data. So the epigenetics doesn't influence what this shows. So that's the trick, and so you have the association between this genetic variant, and I'll explain later why this is sometimes referred to as an instrumental variable. But all it is just your genetic variant. These are a single base pair change variants that influence the amount of production of LDL cholesterol or the amount of LDL cholesterol circulating because of uptake, or any other process which influences your circulating cholesterol level. Now, this can be confounded and there's reverse causation, but this association cannot be confounded. There cannot be conventional confounding. So you have a very simple sum, which is you take this association, and then you divide it by this association, the relationship of the variant to your cholesterol level. That's at the bottom. And then, if this was a continuous outcome, it's just your beta coefficient of this continuous outcome. If this is a categorical outcome, like having a heart attack or no heart attack, then you log it. So you take the log of it in a logistic regression. You get that out of a logistic regression, and you divide the two, and then you get the odds ratio for a given change. Their standard deviation change in LDL cholesterol gives you a 30% higher risk of coronary heart disease. That's those are the sorts of numbers you get out of this. So the basic trick is incredibly simple. And the proof of that is that I sort of developed this, and I'm quite simple, so I wouldn't have been able to think of anything complicated, and I knew rather little genetics. I was interested in public health, and what I was interested in is not wasting money on things that weren't going to work. that's, and so I wasn't interested in genetics per se. I was interested in genetics in terms of what they could tell people about things that could be modified in populations and have health benefits, which very obviously lowering cholesterol has huge health benefits in populations, lowering LDL cholesterol. So that's the trick. And so the arrow becomes, oh, so I meant to go backwards. I can't. Yeah, the arrow goes red because you can start making some sort of causal claims about the association using the genes. Now, the fundamental assumption for being able to make this, the inference that then changing cholesterol will change risk of coronary heart disease, is one of gene-environment equivalence. And that odd term, what that term just means is that it doesn't matter, or it has an equivalent effect, if your cholesterol level is raised, is higher because of the genetic variants you carry compared to other people, or it's higher than the counterfactual if you didn't have those genetic variants. If that's the reason that your cholesterol level is higher at the group level, the downstream effect of that difference in cholesterol is the same as if you have higher cholesterol because you eat a vast number of sausages. So the downstream effect of the exposure is identical in situations where it is the counterfactual genetic change which induces the difference in the exposure, or the counterfactual, or eating a lot of sausages. If that assumption is reasonable and you can defend that assumption, then you can start thinking that this would mean that if you lower cholesterol by targeting those genes, and I'll come back to that in the case of cholesterol, then you would have the same benefit as if you lowered cholesterol by changing food consumption that influence cholesterol level. Now, in what's called the monogenic disease world, this is where you have rare, large effect variants which generate diseases which people will have to some extent or other if they carry those variants. There's huge numbers of examples of when this is obviously the case. I mean, including there are mutations in the LDL receptor which lead to familial hypercholesterolemia, where people have very high cholesterol and great and highly elevated risks of coronary heart disease. But an example I like, because I was interested in the condition, was pellagra, which is a dietary deficiency-induced condition that is caused by lack of niacin, vitamin B3. Which causes this disease where you have dermatitis, you have diarrhea, you have what's sometimes referred to as dementia, but it's not dementia that we've been discussing for the last few days. It's more like delirium. It's a short onset thing that is generated by the nutritional deficiency. The differential diagnosis for that is a condition known as Hartnup disorder, which is a monogenic, a single gene, major effect gene, which stops tryptophan absorption, and tryptophan is converted to niacin. And that has lots of its effects through that. If you're a doctor and you're a very well-trained doctor, like I'm sure Henrik is, for example, if you saw someone with this condition, Hartnup's disease would be rare, but it would be part of the differential diagnosis for a child presenting with pellagra. So we see there that it doesn't, that the niacin deficiency, because if your diet completely lacks niacin, or you have this deficiency in niacin because of the genetic change, they produce the same condition. And in developmental genetics, there's this term gene-environment exchange equivalence or gene-environment interchangeability. Mary Jane West Eberhardt's brilliant text on developmental genetics describes them in these terms. It doesn't matter whether gene expression is changed by something happening externally in the environment, or gene expression is different because you've got a genetic change. The downstream effects on development or disease are the same. And in fact, Zucker-Candle and Villint went as far as saying in 1988, no doubt all environmental effects can be mimicked by one or several mutations. Now, I think for endogenous disease processes, this is almost certainly true. I do think that for some conditions, like being run over by a tuk-tuk, I don't think there's any genetic variants which would mimic being run over by a truck. But for most, for the disease processes that are developing, I think it's the case. And and one example where it's very obviously the case is in the example of LDL cholesterol. So gene-environment exchangeability would mean that the effects of the cholesterol, whether influenced by genetic variation or influenced by dietary differences or cholesterol-lowering medications, then the downstream effect would be the same. And we know this is definitely the case for LDL cholesterol because there are many genes which are directly targeted by cholesterol-lowering drugs. The drugs just simply inhibit the genes, and the genetic variation in the genes influences their level of expression. The one that everyone will know would be HMG-CoA reductase and statins. Statins work because they inhibit HMG-CoA reductase production. There is genetic variation in the HMG-CoA reductase gene. The gene has the same name as the drug, if you like, that influences cholesterol levels. Okay, so that's statins and HMG-CoA reductase. There's also PCSK9, which is a genetic variant which is related to cholesterol level, and there's PCSK9 inhibitors, which lower cholesterol. There's cholesterol ester transfer protein, and there's drugs that CETP inhibitors. And there's Niemann-Pick C1-like-1, which is a gene related to cholesterol uptake across the gut, and a drug like ezetimibe, which inhibits that single gene. So you have many of these cases where the drugs are simply targeting single genes that influence the exposure. And there's been lots of drug trials which have gone on. Generally, the average length of drug trial is about 5 years, a coronary heart disease prevention trial. And you can then map the extent to which the trials and genetic variation influence your LDL. So non-HDL is put here. Non-HDL means the cholesterol, which is low-density lipoprotein cholesterol, but also remnant cholesterol, which is also atherogenic, which is it's the very low density and independent intermediate density lipoprotein. So overall, non-HDL cholesterol, the extent to which the genetic variants influence the LDL cholesterol and the degree to which coronary risk is lower is just sort of linear. The percent risk reduction for coronary heart disease risk. Now, the drug trials, they show the same slope exactly, but they only show about 35 to 40% of the predicted effect. Now, I said something earlier that we should explain why that is. Why are the drug trials not producing 100% of the effects predicted from the genes? So I said the drug trials last 5 years. How long do your genes last? So your genes last from conception. So if you've got genetic variation related to differences in the cholesterol level, you've got that from birth. Atherosclerosis, we heard, 19-year-olds already have atherosclerosis. The disease process is going on for your lifetime. So what the genetic studies estimate is what the effects of if you had a lifetime reduction in LDL cholesterol. You obviously wouldn't expect to see that achieved by just sort of 5 or 6 years in trials. So when thinking about gene-environment equivalence, you've got to say, well, they're equivalent in terms of short-term effects, but the genes, in the case of the drugs, the genes have acted for life, so you're looking at the effects of a lifetime difference of exposure. Whereas the drugs are just 5 or 6 years. And so the longer you take the drugs, by the way, the difference moves closer to the genetically predicted difference. But that's a situation where gene-environment equivalence certainly works. And for any Mendelian randomization study, you've got to be able to justify that the gene-environment equivalence is reasonable. And it relates to biological processes that are influenced by genetic variation. So if you consider body mass index as a phenotype, so the first genetic variant, common genetic variant related to body mass index is FTO, with the largest effect. And that FTO influences calorie intake. The FTO variant, as Nick Timpson showed from the ALSPAC study and many other people have shown since, relates to a very small difference in calorie intake, but one which over a lifetime is enough to sort of generate the differences in body mass index. Variants related to physical activity, then that would be something that would be equivalent with how you would try and target higher body mass index obesity levels. Now, in the causal inference field, there's often this sort of joke that Miguel Hernan made about thinking about how measures relate to outcomes. We measure body mass index in all of our studies, and higher body mass index, higher risk of coronary heart disease, until very low levels, actually, if you use Mendelian randomization appropriately. One way of reducing your body mass index is to chop off your leg. If you chopped off your leg, that would reduce your body mass index, but it's not going to reduce your coronary heart disease risk. Now, luckily, there aren't any genetic variants for losing a leg, but they are genetic variants that relate to heaviness of smoking. There's a genetic variant of the nicotine receptor, which means that if you are a smoker, you smoke more heavily, you find it harder to give up smoking, your cotinine levels are higher compared to the non-carriers, even if you smoke exactly 20 cigarettes a day, because you're inhaling more, you smoke further down the butt, etc. Now, those genetic variants are indeed related to lower BMI, because smoking puts down your body mass index. I mean, one of the reasons people smoke, some people smoke, is because it does lower your BMI. GLP-1 agonists are better, honestly, better way to lower body mass index. But the thing is, so there's genetic variants in the CHRNA5 gene which relate to lower body mass index. Now, obviously, you would not use those genetic variants in studies of BMI, or so you would think. But people do. These sorts of variants are included in Mendelian randomization studies. When I come up to say about how terrible many of the studies are, that's precisely for that sort of reason. If you think about gene-environment equivalence, it becomes obvious that you shouldn't be including genetic variants that are primarily working through smoking, etc. So that's why this is the fundamental assumption of Mendelian randomization is of gene-environment equivalence. So I'm going to give an example which was a sort of pin-up for Mendelian randomization early on. And this was of a protein called C-reactive protein, circulating protein. C-reactive protein, as its name implies, is an acute phase reactant. If you get ill, your C-reactive protein goes up. It's very widely used clinically to say, is this person likely to have infection, or have they, etc. The C in the name is from the capsule of the pneumococcus, because it was identified as something which bound to the capsule it reacted to the capsule of the pneumococcus. But it reacts to everything. If you smoke, your C-reactive protein goes up. Smoking puts up C-reactive protein. So in the late 1990s, early 2000s, C-reactive protein was being considered to be a cause of coronary heart disease because the observational associations were so strong. In some circles, it was being called the new cholesterol, and drug companies were developing drugs to inhibit C-reactive protein levels because people thought the evidence was so strong, as they had done about vitamin E, HDL cholesterol, et cetera, et cetera, et cetera. So this was a pooling project. Many people came together this got data from many studies. This was pre-UK Biobank days, so you had to put together lots of little studies rather than just one big study with 500,000 people. But they looked at a standard deviation higher C-reactive protein just based on a single measurement, even though it's highly variable. So if you had repeat measures, you'd show much larger effect sizes. The effect size would be bigger, obviously, because you remove the measurement error and the variability in something. You'll obviously, if you could measure it every day, you would obviously get stronger associations than if you just measure it once. But it's 50% higher risk of coronary heart disease. And then adjusting for all the potential confounding factors that we could think of, that 50% higher was reduced to a third higher, so half to a third. That's not, but massively, massively statistically robust effect. And based on that sort of evidence, things were going ahead. Now, this is C-reactive protein and confounders. I said everything bad is related to higher CRP, age, higher body mass index, higher blood pressure, etc., right the way down to smoking, lower socioeconomic position. Everything bad was associated with higher CRP. Things that were considered to be good at the time, like HDL cholesterol, were associated with lower CRP, and albumin, higher albumin predicts lower mortality. So all of those confounders relate to your measured C-reactive protein, and all of those were adjusted for in the analysis I showed. Now this is the same thing, but rather than looking at measured CRP, this is a genetic variant in CRP. So this is a cis variant, promoter region variant in CRP, which leads to higher, on average, circulating CRP. So if you grouped people into two groups, with or without carrying that variant, you had groups which had a difference. The groups had a difference in CRP. Just like if you randomized in a trial, you get a group level difference, and that allows you to see whether at the group level it relates to lower risk of coronary heart disease. And I showed a lot of data showing that if you do that for LDL cholesterol, you get the answer that LDL cholesterol increases heart disease risk. So it works. And this just, when we looked at this just looked a bit too good to me, but this is how it was. Mendelian randomization, when it is done properly, does exactly what it says on the tin. It is equivalent to randomization. The genotype does not relate to any of those variables, any of those potential confounders. The genotype is unrelated to the confounder. So the difference in disease risk between groups defined by genotype, is equivalent to what you see in a randomized trial, and it's equivalent to an intention to treat analysis in a randomized controlled trial. And with this build-up, you'll be unsurprised to see the findings of the Mendelian randomization was no effect. So the first time I ever gave a talk to a drug company on Mendelian randomization was in 2005 when I was invited by the head of genetics in Merck to go to visit Merck in Long Island because Merck was developing a CRP inhibitor. And the genetics guy had come across Nick Timpson's 2005 paper by Nick Timpson on MR and CRP and invited, we gave a talk. And eventually, Merck stopped producing the CRP inhibitor, stopped their research program on the CRP inhibitor because of this evidence. That the MR was suggesting there was not going to be any benefit, which there almost certainly wouldn't have been. And so that did avert possibly vast amounts of money being spent on developing a drug that would not have been effective. But to me, the most that's for CRP is a specific thing for CRP. But I want one thing to remember. Every time you read a paper on the latest dietary factor that's supposed to relate to disease or the latest biomarker that's supposed to relate to disease or whatever, then remember this. This is what a null effect looks like when you adjust for a vast number of confounders. You cannot adjust for reverse causation. You simply can't do it. You can never measure all the potential confounders, and if you do, there's measurement error in them. A relative risk going down from 1.49 to 1.33 is what something which is absolutely null looks like. That is what a null result looks like observationally. And that's why most of the studies, you might as well just forget them. Just not read them. Because they're likely showing these sorts of confounded effects. That is a null result. If you remember one thing from today, remember that top half. That is what a null finding looks like. You show massively statistically significant, a term which should never be used, by the way, but massively statistically significant findings that are completely meaningless. That's a null result. Now, you can do Mendelian randomization, obviously, in both directions. CRP was supposed to cause some cancers. It was definitely supposed to cause type 2 diabetes and possibly obesity. It was being studied investigated for those things. And indeed, there's a huge very powerful observational association between CRP and BMI. And this is the regression of BMI, of BMI standardized differences and CRP. And what you see is that increasing BMI, there's increasing CRP. And then that's the central line, which has no confidence intervals because the sample size is so large you can't see the confidence intervals. But then this line, the line that basically goes perfectly with that, but then with those confidence intervals, that is the Mendelian randomization using the single genetic variant in FTO as a variable for BMI, as the genetic instrument, if you like, for BMI. So what this tells us is that BMI influences CRP. There's a causal effect of higher adiposity on CRP. But that, of course, what that means is that FTO associates with CRP, which I will come back, very heavily, massively genome-wide significant. I'll come back to that. If you do it in the other direction, the observational association is, of course, the same, just on different scale. This is CRP onto BMI, but the Mendelian randomization effect is flat. So there's no causal effect of CRP on BMI. The directional effect is BMI influences CRP. So Mendelian randomization here is saying that BMI is definitely a confounder because it influences CRP and it will influence coronary heart disease, type 2 diabetes, etc., all the things that CRP was supposed to cause. But if you consider gene-environment equivalence in this setting, if you do a GWAS of C-reactive protein, a genome-wide association study of C-reactive protein, FTO is the 4th variant in the list. There's now 100 or more genome-wide significant variants related to CRP. The 4th one is FTO. Obviously, FTO would not satisfy gene-environment equivalence as a genetic variant to look at CRP. It would violate it completely because FTO influences BMI, which then influences CRP. FTO is not equivalent to a monoclonal antibody lowering your CRP. The genetic variant, the cis genetic variant in the CRP gene region that influences higher CRP, oh, sorry, I can't, that we saw, we see there, that is equivalent. It's reasonable to say that would be equivalent to a monoclonal antibody lowering your CRP. FTO obviously would not be. Yet, huge numbers of MR studies now of CRP just take all the genome-wide variants, including FTO, bang them in, and get the wrong answer. So, yeah, when introduced, Mendelian randomization links strictly to Mendelian principles. The Mendelian principles are, for Mendel's laws, are that meiosis randomizes the genetic variant from the parent to offspring. The offspring basically get 50% of alleles from both parents, and it's randomly chosen which of the two parental variants you get. The exception being that you have linkage disequilibrium or linkage from the parent to the offspring, which means that you get a chunk from the parent to offspring. There's a chunk of variants that go with that. But if you have siblings, then between siblings, you are looking at something which is essentially random. And that is analogous to RCTs, and that's what we said was the RC randomized controlled trial analogy is with a parent to offspring study, between sibling study. But there was just simply no data at the time. So we said this terrible diagram showing Mendelian randomization was being from parent to offspring. But certainly in 2003, there were no very large studies hardly at all of genotyped sibling pairs. So we referred to approximate Mendelian randomization as being what you would see at the population level. And I'll come back to why that's important. And we should have carried on calling it approximate Mendelian randomization. AMR, but that would get confused with antimicrobial resistance, but still. When we introduced this, hadn't thought of this as an instrumental variable approach, but what Mendelian randomization is doing in this setting when you're making quantitative estimates is it's treating the genetic variance as something that economists referred to as an instrumental variable. So for an instrumental variable is, in econometrics, this is in general now, not thinking about genetics, but my examples are all genetic. That is a variable which is associated with the exposure. So the IV has to be reliably associated with the exposure of interest. And then the second assumption is that there's no confounding between the instrument and the outcome. That's the second instrumental variable assumption. The first one you can test statistically. The second one, you can't test it. You can only subject it to sensitivity analyses. Now, what could the confounders be in this situation? As I say, you're born with germline genetic variation. So you can't have smoking and et cetera can't be confounders or reverse cause. The confounder can be, a potential confounder is ancestral population, the ancestral population you come from, because some variants have very different allele frequencies in different ancestral populations. There are a relatively limited number of these, certainly at the common variant level, really rather few where there's simple limitation. The best known example of something which is a common variant and shows this is in East Asian populations in China, Japan, Vietnam, etc. There is a genetic variant in acetaldehyde, in aldehyde dehydrogenase 2, ALDH2, which breaks down acetaldehyde, and when people drink alcohol, alcohol is broken down to acetaldehyde. Acetaldehyde gives you all those sort of negative effects of alcohol consumption, like flushing, palpitations, headache, guilt. Actually, not guilt, but it gives you all the other adverse consequences of drinking. And in East Asian populations, I'm sure many people have East Asian friends who don't drink alcohol because if they drink alcohol, they flush and get headache and get palpitations. And if you're homozygous for that variant, of which reasonable proportions are, you really can't drink. It's intolerable. If you're heterozygous, you can drink, but tend to drink less amounts. But that's an example of a variant which in East Asian populations is common. In European populations is literally zero, virtually non-existent. And if there is, there'll be a lineage that will explain that. So now if you have that sort of situation, you can have what's called population stratification, and that is that if you have a mixed European origin, East Asian origin population, say San Francisco, then if you've genotyped people for that variant, then it would appear that that variant causes chopstick use, which obviously it doesn't. That variant is related to chopstick use because of ancestry. So that's the only form of straightforward confounding you can get is with population stratification, is when you have these mixed populations. Now, with genome-wide association data, that is taken into account by creating what are called principal components, which are ancestry related, and you adjust for the ancestry related principal components. So that's why you'll often see in GWASs, in many papers, you'll see you'll see that form of adjustment done to take into account the only real conventional confounder. There are the only other sort of set of potential confounders are referred to as dynastic effects. The one thing that can be said to cause your genotype is your parental genotype. Obviously, your genotype is influenced by your parental genotype because it is 50% of your parental genotype. And your parental genotype might influence your outcomes or your exposures because your parental genotype creates the environment which you grow up in, including your maternal genotype influences your intrauterine environment. So if your mother carries a particular genotype, your first 9 months will be spent influenced by an environment, an intrauterine environment that reflects your mother's genotype. And the only example, really, of family-level proper MR that we had in 2003 related to such dynastic effects And this is the figure from the paper. And it relates, and one could see this, but we're looking at neural tube defects. So, you know, some offspring are born with neural tube defects. And now, if your mother has a genetic variant that's related to lower folate, higher homocysteine, for example, in the methylene tetrahydrofolate reductase gene, MTHFR, there's a famous genetic variation in that gene which leads to lower folate, even though folate hadn't actually been measured in 2003. Homocysteine was measured, which folate influences. And then if your mother is homozygous for that genetic variant, you would have been, your first 9 months would have been spent in a lower folate environment. Your father's genotype obviously wouldn't influence the intrauterine environment, so that's a perfect negative control. And the fetus obviously inherits 50% from mother and 50% of father. Now, the genetic variant related to lower folate in the mothers, if they're homozygous, is related to twofold the risk of neural tube defect. Now randomized controlled trials, of course, show that folate supplementation periconceptually and even in early pregnancy very substantially reduces the risk of neural tube defect. Supplementing the dad will have no effect on neural tube defect. And the fetal genotype probably has no effect. But the fetus, because the fetus has got half of the mother's genotype, you see a raised association. It would be wrong if you saw this person at, if you see the offspring age 10 and they've got a neural tube defect, if you carried out MR, it might appear that it's their folate that influences having a neural tube defect, and you should supplement with folate to reduce that risk in them, which obviously would have no effect. But this is an example of how one's own genotype might be related to your outcomes because it's the parental genotype that actually generates the outcome. And this is important for these developmental issues, but also it might relate to environment more generally. If you think of your parents have genetic variation which is related to drinking more alcohol, smoking more, etc., and you'll grow up in an environment which has been partly created by the parental genotype. Well, parental genotype relates to higher educational ability, and there'll be more books in the house. So you'll have a different environment. So for confounding, there's two possibilities: is population stratification, and the second possibility are these dynastic effects, which tend to relate to either intrauterine environment or behavioral factors. But largely, it does what it says on the tin. The third instrumental variable assumption, again one that can't be tested, is of has this horrible name of the exclusion restriction. This is from econometrics. All that assumption says is that the genetic variant influences the outcome entirely through the exposure. There's no other pathway, which is in genetics is called horizontal pleiotropy, where the genetic variant goes through some other pathway and influences the outcome. Now, genes can't cause anything directly. All genes influence development, health, etc., through phenotypes. So this must be working through some other phenotype that you don't know about. But if there's a horizontally pleiotropic association, then you'll get the wrong answer, or could get the wrong answer. So those are the basic assumptions. And early on, sadly, a lot of people thought, well, you can test this by adjusting for the mediator, and that if you adjust for the mediator, that association, the association of the genetic variant should go away. People started calling this the causal inference test. It got quite well known as called CIT. I'm not going to mention the name of the very famous and extremely well-paid person who works for a pharmaceutical company who owns 2 jets through his pharmaceutical company interests, who introduced this test. Maybe I should have taken a different career choice, but anyway, I didn't. And I got fed up of writing letters saying that this was not the case. I pointed out that economists have been dealing with instrumental variables for at least 50 years. And if someone had solved this, they should have got the Nobel Prize for economics. So either these people were producing things that were worthy of Nobel Prizes, or it was wrong, and it turned out that it was wrong every time. They hadn't. So if you ever see anyone saying they've got a test for the third assumption, it's wrong. But it's actually quite important for general reasoning and also for Mendelian randomization as well, why one understands that adjusting for X would not work. Trivially, there will be measurement error in any measured X. So it wouldn't work because there's measurement error in that. But more importantly, or maybe less well realized, is that adjusting for X will be might well be what's known as adjusting for a collider. And collider bias, which I'm sure many people here will have heard of, it's most easily explained with an example. So let's imagine that you're in the US and you're in a situation where people get into university, as they do in the US, either through their SAT scores, which are standardized academic achievement scores that everyone takes at schools, or through a sporting prowess. You get many US universities have a large number of scholarships for baseball, basketball players, and baseball players, and American football players, etc. So imagine those are the 2 ways into college. Now, in the general population, in the population at large, think that there's no association between academic ability and sporting prowess. So they are completely unrelated in the general population. But then if you go to one of these university campuses, and there's a student, and you sort of throw the ball to the student, and the student sort of tries to catch it, they fall over backwards, and the ball bounces off their head. You can then go up to them, and you can confidently say, "You are very clever." Because there is no possibility they are there because of sporting prowess. And in that college, the correlation of academic ability and sporting prowess is minus 0.6. So you generate, if you adjust for something, you generate massive amounts of correlation and confounding, even when in the general population there's none at all. So if you try the Nobel Prize worthy approach of adjusting for X, everything else which influences X, everything which influences smoking or cholesterol level or whatever this variable is, becomes related to your genotype. So you generate confounding when it doesn't exist. But there's so many. So that's why these sort of things where you see that people just say we adjust for everything, is one's got to think really carefully about the possibility of introducing collider bias through adjusting, through adjustment. So that's collider bias. Now, now moving just into genetics. So in genetics, this is horizontal pleiotropy, which is working through an unknown exposure. That's the main problem in Mendelian randomization. There's also a form of pleiotropy which is called vertical pleiotropy. And vertical pleiotropy, the genetic variant influences the exposure, and the exposure influences the outcome. So that is like a genetic variant that influences body mass index, like FTO influences body mass index, and then body mass index influences coronary heart disease. Now, you will recognize very easily that is simply Mendelian randomization. So Mendelian randomization is just the use of vertical pleiotropy for making making causal inference. You can have mediated vertical pleiotropy when the variant influences some upstream phenotype which influences X. That's okay, but that's the situation you're often in when you do a GWAS study. You find that the genetic variant is associated strongly with X, but it's clearly not a direct association, that it's working through some other phenotype. So every time you don't know about that phenotype, you're making the assumption that this unknown phenotype doesn't influence Y. So imagine you're in a situation where you didn't realize that FTO influenced BMI, but then BMI influenced CRP, and you used FTO as a genetic instrument for CRP in ignorance of the BMI effect. Then you get the wrong answer, which you're misspecifying the primary phenotype. The primary phenotype is actually BMI, but you're saying that actually is a genetic variant for CRP. And that, you get the disastrously wrong answer. So
is that a common problem? It feels like it could be a common
it's
problem.
extremely common problem, yeah. And it's why we have the disaster of the number of terrible Mendelian randomization papers today.
Okay, so
that's why this is why I'm going through all this. I thought walking here, it's why it's a good thing walking. I thought I should go through these things in real detail, and it doesn't matter if I don't get to down the line. And I've just noticed that I've already been speaking for an hour. So I just want to say that I fully understand that people have homes to go to, places, other things to do, and I honestly, if you've got other things to do, do go. But I will carry on, assuming people are interested. I'll carry on this because it's a major problem. Yeah. But I fully understand if people have other things to do, and my email, and Nidhi can give people my email address. I'm very happy to answer questions on email, etc. But I'll carry on. So yeah. So and now you know this common problem, this major problem. There's a simple solution if you have some idea about about what this upstream phenotype can be. Is you use a method which is called Steiger filtering. And what Steiger filtering does is very simple. You just look at the variance explained in BMI and CRP, and if the genetic variant is working through BMI, it will explain more of the variance in BMI than it does in CRP. And so when you use Steiger filtering, you exclude any variant which explains more of the variance than anything else. A genetic variant, which is a reliable instrument for the exposure, should explain more of the variance, or pseudo-variance if it's a categorical outcome or categorical measure. It should explain more of the variance in your exposure of interest than it does in anything else. So in Steiger filtering, it works very well if you have some notion of what the exposures are, or you can just do it against everything. And we've got a preprint out showing about how beneficial this is and how it gets rid of lots of the nonsense results. Because if you have this situation like FTO, BMI, CRP, BMI causes everything, then you have a situation which is called correlated pleiotropy, because the pleiotropic effects correlate perfectly with your genetic variant wrong exposure effects. Your genetic variant CRP effect will correlate with your genetic variant coronary heart disease effect perfectly. And virtually many of the sensitivity analyses that have been introduced cannot cope with correlated pleiotropy. So, and it's correlated pleiotropy which is causing many of the problems in the use of MR, and if thought through in this framework, can be dealt with. So I mentioned the estimates that you get for cholesterol and coronary heart disease, for example, or CRP and coronary heart disease, which is null in that case. To interpret those estimates, you've got to think about the exposure period, that genetic variance relates to exposure right the way across life, from conception on, and the sorts of things you're wanting to do environmentally will often relate to an intervention in people who are 50 years old. So you've got to consider the exposure period. Might relate to an exposure during the critical period. So if your vitamin D levels when you're an adolescent influence your risk of multiple sclerosis, which they very might well do, then of course the Mendelian randomization will show an association with with vitamin D at age 50, but giving vitamin D at that age will be too late. So you've got to realize that the genetic variants influence the exposure right the way across life, and there might be a critical period. And most importantly, generally relates to disease occurrence, not the outcome of disease progression. Because what we're often interested in is treatment. So for treatment, people have already got the disease, and then you want to treat them. For many conditions, the factors that influence incidence are very different from those that will influence progression. Earlier, a couple of days ago, when I mentioning this, I mentioned smoking and lung cancer. And the genetic variant that I talked about earlier about heaviness of smoking, of course, that relates very strongly to lung cancer. It's the top genome-wide association study hit for lung cancer is that genetic variant, because it relates to heaviness of smoking. So at the cost of $10 million, Mendelian randomization did indeed prove that smoking causes lung cancer, but we knew that already. But once you've got lung cancer, stopping smoking isn't useful. So if you want to know about disease progression and treatments, then you need a group of people who've got the disease, and then you've got a lot of follow-up data on them, and you've got to do Mendelian randomization on disease progression. But you've got to take into account the fact that you are conditioning on a collider. Getting disease is something that you are automatically conditioning on in that analysis. Everyone has to have disease. So you need to use a whole suite of methods that have been developed to take into account that collider bias. And if you don't, you can get the wrong answers, especially in relation to variants that cause disease, that cause the disease in the first place. So there's been many wrong answers demonstrated on disease progression without taking that into account. And there's a paper by Ruth Mitchell in PLOS Genetics a couple of years ago which reviews all of the approaches to dealing with that. Now, there's a massive industry in sensitivity analyses for Mendelian randomization, which I'm not going to go through. But these are the sorts of these are if you're actually performing MR analyses, then these are the ones you would want to know about this. So let's take a relatively simple example of some Mendelian randomization that is of some importance because it's related to a treatable condition, which is osteoporosis, fracture risk, etc. And that relates to a protein called sclerostin. Now, sclerostin inhibition increases bone mineral density. This is a nice example because sclerostin exemplifies genetically based drug discovery because sclerostin was identified in monogenic conditions where people had very high bone mineral density and had no sclerostin. It was sclerostin knockouts. These people had massively high bone mineral density. So through that, it was realized that a monoclonal antibody to sclerostin increases bone mineral density. And randomized trials do indeed show that monoclonal increases bone mineral density and reduces fracture risk quite substantially. So it's genetically driven drug discovery. Now, there are genetic variants that relate to sclerostin. As I said, everything is heritable. And if you do a GWAS of sclerostin, this was the first one, a couple of genetic variants were found that were very reliably related to sclerostin. In a couple of genes that were sort of reasonable to think that those genes might influence sclerostin in a way that was interpretable in terms of how modifying it would be. And the Mendelian randomization did suggest that that sclerostin itself lowered bone mineral density and increased fracture risk. So the Mendelian randomization showed the same as was shown in the randomized controlled trial. Now, around the second round of GWAS was done, and this still the big element was the two hits we'd had earlier. But this time we were very pleased to get there was a cis variant. It was a relatively low frequency cis variant, but this was a variant in the promoter region of the gene SOST, which produces sclerostin. So this is similar to the CRP situation, and we sort of felt, we felt better about this, that this was very likely to satisfy gene-environment equivalence. But when thinking about gene-environment equivalence, you've got to remember that sclerostin is produced by osteocytes and osteocyte-derived cells in bone, and it's also produced in what are called osteocyte-like cells in the coronary arteries. Those are the two large main places it's produced. The sclerostin circulating in the blood is that's sort of leaked out of the bone or out of the etc. So blood, the circulating sclerostin, which is what the biomarker people are always measuring, isn't the actual target. When you're giving the monoclonal, what you're hoping is the monoclonal is acting in the bone to stop the sclerostin thinning your bones. That's the hope. So the SOST variant is the one that's really likely to be equivalent to the local production of sclerostin. And indeed, the SOST variant, for the same level of blood sclerostin, the SOST variant showed a much larger effect, which makes absolute sense given the thinking about gene-environment equivalence. But our meta-analysis, so this GWAS also showed lots of the problems with Mendelian randomization that I've mentioned. In particular, a large number of the variants were variants that primarily related to bone mineral density. And if you didn't do the Steiger filtering, they explain more of the variants in bone mineral density than they did in sclerostin. And these variants are very likely to be having the effect because biology is homeostatic. We're in a situation where everything is regulated, has feedback regulations. So increased bone mineral density will lead to increased sclerostin levels, which are trying to downregulate the bone mineral density. So the reason why the bone mineral density variants relate to sclerostin will be because of this downregulation. And indeed, it's the higher bone mineral density variants related to higher sclerostin. If you use those variants, you naively use those variants as instruments for sclerostin levels, you got precisely the wrong answer. It appeared that sclerostin was actually increasing bone mineral density. So just throwing in all the variants is terrible, can be terrible. And these are the results with sclerostin and bone mineral density showing that of the variants which survived Steiger filtering, they all showed effects in the same direction. This is the predicted effect of a given difference in blood sclerostin, because there's no measures of bone sclerostin, but the cis variant has a much stronger effect, which is what you would anticipate, because the cis variant is related to the local bone level of sclerostin production. It's the strongest instrument. So I'm going to skip a load of slides. I'm going to skip a load of slides now, which are there examples of MR that I like because it's the sort of area I'm working on. But I'm not going to bore you with another example. But so I've shown some examples, I hope, which show interesting findings. So I'm going to move on to how it really is. So I think I've been foreshadowing the fact that if it's done badly, Mendelian randomization produces silly results, and there's a massive number of these studies. So the types of MR study when originally done were based basically on individual data on the outcome and individual data on the exposure. But now this isn't the case at all. Most of the studies done are based on freely available summary level data on the exposure. This is GWAS's, genome-wide association studies that have been done of exposures, and these are deposited in catalogs, including probably the most used is our IEU Open GWAS catalog, so you can just easily go to that catalog and get those findings. And the summary GWAS data on the outcome, which can also be taken. And in 5 minutes, you can calculate the causal effects. So I call these summary data exposure, summary data outcome, I call noodles for reasons I will come to. The other type of MR study is where you have the individual level data on the exposure and the outcome, and that is one sample MR, and that is fine unless you're getting both, unless you're actually generating the instruments within the same data set as you're applying them. And if you do that, you get a situation known as overfitting, because just random fluctuations will get picked up, that associate things will get picked up, and the data will reproduce those chance associations. You can easily deal with that with the jackknife approach, whereby you split your sample, like UK Biobank, if you're analyzing UK Biobank, you split it into into 10 groups. You GWAS 90% of them, of the group for the exposure, and then you apply that to the outcome in 10%, and you get an estimate. And then you repeat that nine more times, and then you meta-analyze the 10 effects. You lose very little statistical power, and it is protected from overfitting because you're not including the same data for any of the actual pairing of exposure and outcome. So there's a paper by C. Fang a couple of years ago where we've released software for doing those sorts of jackknife analyses. Probably now, there's a hybrid sort of design now which is used a lot, which is you take summary data on the exposure, not from UK Biobank, so to avoid overfitting, and then you use the biobank outcome data. So, and then Million Vets program and other very large biobanks feed into this. And that combination is the unicorn. That's a rare, very rare do you see that combination. But this is Garcia's Chronicle of a Death Foretold. So when two-sample MR took off, we wrote a paper in 2016 saying that it's too easy. There's problems with the GWASs that are getting upstream variant effects that might be spurious. There's also just technical problems of palindromic SNPs, which are difficult to resolve. And we used one paper as an example of this, and that led to the first retraction. That paper was then retracted because of the silly things it had done. But you can't police the literature by retraction at a time. And every single Mendelian every single analysis based on IEU Open GWAS has been done. So when I get asked to review any two-sample MR study, I refuse to review them, and I say to the editor, the results are already available on Epigraph DB on its database, where you can go and find any two-sample MR results. So it's a meaningless paper. Now, but what's been seen recently is this. These are the numbers of papers. These are papers of Mendelian randomization in their title. This is per week. This is carried on going up. This is from a few months ago. This is a true exponential. By 2029, at this rate, every single scientific paper in the entire world will be a Mendelian randomization paper if this carries on. It's good for my citations, but it's terrible for the discipline. And the problem is there's lots of extremely good Mendelian randomization that can be done when it's triangulated, put together with other data, etc. But just simple standalone two-sample MR papers are just often wrong and nonsense. And I call these noodles, no-nulls, and numbskulls, 3 N's, to remember what these types of papers are. Now, the noodles it's sort of unfair, but this is because the paper on noodle intake and metabolic syndrome got picked up on Twitter in the days when it wasn't the sewer that it is today. It was about 2 or 3 years ago. And they got very widely tweeted, and everyone was sort of taking the piss out of this paper. Now, the paper was ridiculous because there is no way you could just actually have some genetic variant which plausibly influenced noodle intake separate of other forms of dietary choices and body mass index and etc. things that influence noodle intake. You just can't isolate noodle intake as an exposure using genes. The actual paper it was actually technically well done. The people realized about overfitting and handling of the genetic variants. There were no palindromic SNP problems, etc. It was difficult so you couldn't say we should retract it like the earlier paper, but it was just not a possible thing. And you get causal effect of COVID-19 on cancer risk. The outcome data was all before 2019. There was no COVID-19 around when those outcomes occurred, so how could you be looking at COVID-19 risk? This lipid-lowering therapy, this GWAS, which statin people were on in UK Biobank, whether it was atorvastatin or simvastatin. I mean, you were basically looking at the genetics of the physician who was prescribing the drug to see whether they were up with the literature or not and with the drug they were giving. You were saying nothing about the patient by doing this, but you can do it. The problem is you can do all of these things. Or air pollution. This is based on, believe it or not, you GWAS their postcode, and then you assign the air pollution level from around their house based on the postcode. It's rather difficult to think that where someone lives, the only effect on health outcomes are through air pollution. Anyway, so they're ridiculous. This is translated by Xifang from the Chinese website. This is where you can pay for them to help you produce a paper. So in the Chinese system, you have to have papers that are in journals with such an impact factor level to get a job. And so there's a vast industry in this. They advertise straightforwardly that we got this paper. Our company got a paper in a journal for someone. The authors are nothing. The authors buy the paper. With a 15.3 impact factor. That's a bit of a silly thing to say because you could easily identify the exact paper that it had to be. And you can save money by buying 5. The cost was 3,398 yen per single paper, and if you bought 5, you got 1 free. And when you got 4 for the price of 5, you got 5 for the price of 4. And then when a new exposure comes out, then there's things that haven't been looked at so far. What you get is this flood of papers all appearing within weeks of each other. The implausible thing here is the gut microbiota influencing erectile dysfunction. And believe it or not, look at the titles. This is exactly the same thing. Now, some of these journals are reasonable journals. They're not all nonsense journals. Genetically intestinal. a lot of people are interested in the gut microbiota and erectile dysfunction. I mean, they're just showing the same things. Those are noodles. Now, I mean, you know, maybe they're not having any influence. Maybe they are just nonsense, but they are nonsense. And it's obviously detrimental to how Mendelian randomization is seen and how it can be applied. The no nulls is really important. Is the most influential Mendelian randomization work has been nulls. Most things do not cause most other things. And so the Mendelian randomization on HDL cholesterol, C-reactive protein, fibrinogen, activated lipoproteinase, which was being, drug trials were done which failed. And many of these other things, it was these null findings that were key, that were really important. They were really important nulls. You don't see nulls don't appear anymore because no one's going to pay for a null paper. So there's noodles, no nulls, and then the numbskulls, which in a way are the most damaging, are ones when methods, sort of new complex methods which neither the people who've developed the methods or have applied them actually really understand appear, and they then flood the literature and they get into decent journals. I call them numbskulls because they just make your head hurt trying to work out. It's a lot of work working out why the methods are wrong. I mentioned nulls, and one of the really important nulls has been on things like vitamin D. I know I started by talking about all these trials of vitamin D which showed no effects despite the observational data being very strong. And this is a very good systematic review of vitamin D MR. But when it was saying that everything's basically null, apart from multiple sclerosis, which stands out as strong, was that when it was submitted, it had all these numbskull nonlinear papers, and it claimed effects of vitamin D, but the authors went back and completely redid it when this was pointed out to them at review. And so this is in one of the Lancet journals, a paper now saying that vitamin D was protective of coronary heart disease, and it developed a nonlinear MR approach whereby you could apparently, the authors claimed, look at the shape of the association rather than just the overall effect of vitamin D. You could look within strata of vitamin D. And these are the observational data in UK Biobank on vitamin D and all-cause mortality. You saw the same for many other causes. Whenever you see the same shape of association for all causes of mortality, you think this is unlikely to be causal because causality doesn't work at a general level. So this will include accidental and violent deaths, etc. You think there's confounding going on. And this is a figure that appeared in that paper. The paper had 100 authors on it, including leading people in the cardiovascular field. But these are the 4 strata of vitamin D at baseline, from deficient, insufficient, sufficient, and adequate. These were the strata. And they were predicting these beneficial effects to greater or lesser degree overall in every strata. And yet, overall, when they just looked at the whole population, there was a null effect. So in every strata, you're getting lower risk, reduced risk that's greater than the overall absolute null. Now that seems implausible, and indeed it is not just implausible; it is absolutely impossible, and a sort of vaguely remembered sense of having been told about Simpson's paradox, unfortunately, doesn't save it. That is just literally impossible. That cannot be true. But it was published in one of the Lancet journals, and it was just, MR simply recapitulates confounded observational findings. The shape of this association, if you've got to turn it that way up, is precisely what you see in the observational data. It's that curve like that. So that's very sad. we ran precisely the same analysis as the authors had done on what's called a negative control. This is something which cannot be influenced by the exposure. So we took that's the association repeated by a new approach which fails to show it. No, sorry, this is what I was talking about. We looked at chromosomal sex, and vitamin D changed your chromosomal sex, and vitamin D made you younger. Now, this is not actually possible. And if you do these negative control analyses, can be really informative because they provide a sanity check on whether anything can be true. And sorry, I'm going the wrong way. And the paper was retracted. The people, Brent Richards, who'd written a commentary on it, they retracted their commentary even. So, but it's an incredible amount of work to get these things, especially from a Lancet journal, because the Lancet does not like to have to retract papers, even when you could actually say this is literally impossible. But eventually, they retracted the paper, and the whole stack of these nonlinear papers have been retracted. The nonlinear papers claim to show that HDL cholesterol was protective after all, which is a little bit sad, because seeing as that was a really major finding from MR. And the reason for this, I've already really gone through this, I think, at length, is just that the increasing size of your GWAS mean that you're just picking up more and more variants which are upstream of the exposure and affect the outcome. So I showed you this, this is the well done MR with the cis variant and CRP, showing that the variant relates to CRP but is unassociated with all confounders. If you just take the full GWAS for CRP, you get this. So the CRP instrument now relates to smoking, body mass index, because FTO is picked up. Smoking is picked up because smoking puts your CRP up. Blood pressure, waist-hip ratio. So these are all the same confounders as we looked at earlier and weren't related, and now many of them become strongly related. If you look at this, only 2 or 3 of them are not P less than 0.05 related. So you just bring back all the confounding back into the study. So if you consider CRP as the phenotype for gene-environment equivalence, the cis CRP variants work. The cis CRP variant would be equivalent to a monoclonal in terms of the influence on circulating CRP level. Body mass index variants clearly aren't. Smoking variants clearly aren't. IL-6 variants absolutely definitely aren't, because IL-6 influences lots of outcomes. And an anonymous trans variant that you don't know what it does is very unlikely to be a reliable instrument for MR studies. So this is why I started with saying that the fundamental principle, and if you're going to work in Mendelian randomization, the fundamental principle is gene environment equivalence. And in multivariable MR, you need to consider multivariable gene environment equivalence, which is I won't go into. So what's to be done? Journals shouldn't send out to review 2-sample studies. Reviewers should refuse to review such. The journals shouldn't send out to review any MR papers even without there's a thing called strobe MR that was published in JAMA and BMJ, which is about what stuff has to be reported in any MR paper, and the checklist should be reviewed, and the methods should be definitely much more rigorously evaluated before implementation. There's just huge numbers of papers turning out methods which literally the authors and the developers don't understand what they do, but they look good. So I'll honestly only do 4 more minutes on what it should become. Firstly, it should be presented within a triangulation of evidence framework. So you should, the MR data should be put together with data from other approaches to addressing the same question, from CRISPR experiments, animal studies, right the way through to different observational approaches. Reconnect with biology and cease to be just mechanical application of IV. Contribute to the collection of appropriate data. The thing which is really missing is our studies of disease progression. That's if you want to develop drugs which can actually help treatment after disease development, then you need to have cohorts of disease progression data, and you need to apply the appropriate methods for case-only analysis. Should be applied to questions of natural selection. So you've got to remember that evolution is through phenotype. Genes are not selected for. What can be selected for and influence selection is the phenotype, and we recently used MR in a paper, Nature paper on lactase persistence, which tells you that anything you've learned about lactase persistence is not true. It hardly relates to milk consumption, and the reason it was selected for was because of infection. But anyway, quite an interesting story. Should be applied to non-human life forms. So sadly, our veterinarian from yesterday isn't here because there's lots of MR being done in the economically important areas like milk production, like rice yield. So I love seeing these papers, and we've set up a consortium called Mr. Mouse, which is Mendelian randomization in mice, in outbred mice crosses, which is really interesting. Be used to understand how confounding generates robust and non-causal associations. I showed that FTO influences CRP, so you can just extend that through BMI. You can just extend that by looking at all potential confounders. So for something like the noodles situation, where you can't do MR on noodles, what you can, though, do is look at upstream traits, which you can use MR with noodle consumption as the outcome. So and then you can show that BMI influences that, educational level influences it, smoking influences it, et cetera, whatever. And then you can use that to say how much confounding is there and what association would you expect to see. So you don't need to just sort of throw up your hands and say, oh, the genetics aren't useful because I can't get good instruments. The genetics can be really useful for examining why you are getting associations, apparently replicable associations, and there's a lot of work going on in that area. And be combined with studies of somatic mutation for a combined Mendelian and Darwinian approach to disease biology. This is using MR to look at cancer promoters. And thanks to people who do all the work, I can't do any analyses anymore. I'm de-skilled in terms of, as I said yesterday, I'm very happy to talk to anyone who's doing Mendelian randomization, but I'm not going to look at your code. I'm being honest here. This is it's a primer which really goes through the basics right the way through to the sort of correlated pleiotropy approaches. This is a rant about how terrible things became with some of this overproduction. And then finally, this is in case if there's, everyone has in their lives people who you've got to buy presents for them, just their sort of family, friends, people you feel you have to buy presents for them, but you don't like them that much. I'd like to recommend this book, Combining Human Genetics and Causal Inference to Understand Human Disease and Development. The ideal present for someone that you don't like that much. Thank you very much.
Thank you, Professor
Thanks for your
Judge.
patience with me going on an hour and a half. Sorry. There's a hand up there.
Hello, thank you for the insightful talk. It was wonderful. I don't see any example of yours in all of the talk about the infectious diseases.
Yeah.
Now, India having a lot of them, so is that the MR alone cannot work? And if triangulation has to be
Yeah.
taken into consideration, then what method of triangulation would you suggest?
That's really good question, and it's one that I'm very interested in because my main interest is switched to infection over the last few years. So there are very good examples of MR and infectious outcomes when looking at cofactors. So the examples here are of Burkitt's lymphoma, which requires early life Epstein-Barr virus infection, but you only see Burkitt's lymphoma in malarial holoendemic regions for malaria. And so studies have been done using sickle cell as an instrument for falciparum malaria. You know that everyone has had EBV by the age of 2. So in the Uganda general population that we work with, literally all the kids at 2 or 3 have infection. And once they've been infected, you know that they carry the virus forever. And you show very strong effects of sickle cell. So, I mean, now that is an example of triangulation because Burkitt, I haven't got, if it's my laptop, I would put up the photograph of Burkitt's original map that I took off the wall of the Uganda Viral Research Institute where Burkitt studied it, his actual map of mapping the cases to, it was beautiful epidemiology. You know, he mapped the cases to regions and showed that you only saw Burkitt's in malarial holoendemic regions. So there are examples like that when you can look at cofactors, and work is ongoing with in relation using the same sort of approaches in relation to EBV and multiple sclerosis. Now the second thing is looking at quantified antibody levels and the many different antibodies. So with EBV, there's about 15 antibodies that are measurable, and people think that some of these antibodies might actually relate to the disease activity. You can GWAS. You can begin to GWAS factors that actually influence them. Given you've been infected, the actual levels of quantified antibody. I mean, and the thoughts with EBV, for example, are that the infectious dose might sort of set the proportion of the beta cells in which the EBV viron has been incorporated, which will be reflected in certain of the antibodies. Now, if you actually look at the levels of antibody, the interesting thing is, of course, as the sensitivity of these tests gets higher and higher and higher and higher, what you get is clearly, in some people who have not been infected, they have some sort of antibody signal, and you have a bimodal distribution of the antibodies. Now, the cutoff point for infection, people generally draw it by eye. So, but we've been working on a nonlinear method, which works, which we actually invented because of analysing why the nonlinear MR had got the wrong answers. We found out about interactions and realising that you can actually infer what you can actually. Firstly, you can get the right curves and calibrate, which is important, but that's not an MR issue. But then you can actually look at factors that relate to antibody level given infection, and so which might tell you something about infectious dose. So in the Uganda data, Kaposi's sarcoma herpes virus is really common in Uganda. I mean, it's not like EBV. It isn't that 100% of kids get it eventually, but a large proportion of people get it eventually. And you can show that really early age of infection relates to higher antibody levels later. And then the MR can help tell you, but is that then going to actually influence disease risk? there's also a lot of work going on with now, of course, HLA. Obviously, if you GWAS just being infected or not, you're guaranteed to get HLA. You get hits in HLA. I mean, and they're very difficult to interpret. There's not genetic co-localization methods that work, but there's a lot of development going on co-localization approaches. But you can never. But the HLA is likely to influence to have pleiotropic influences, but at least you can start to sort of understand. You can start to understand those. I mean, the problem is that, and you'll, the moment I mention it, you'll know what the answer is of course, UK Biobank released data on 10,000 people with 30 or 40 antibody tests. There's hundreds of ridiculous two-sample MR papers appearing saying about this infection and that, just having GWAS 1-0 infected or not, and UK Biobank and thinking it was transferable and useful, and most of the HLA hits, which is clearly pleiotropic, etc. But no, so there are ways of using the sort of MR approaches and genetics to help with infection. They're really in early stages of development. That's what I'm going to spend the rest of my career on. I'm retirement age. I've got my pension in the UK, but and the area that I'm most interested in. It's one of the areas where I really think the genetics can really help, but not in the very simple instrumental variable sort of MR approaches. But the genetics are going to help. Yeah.
Hi, Professor. Thank you for explaining various aspects of Mendelian randomization. So I was thinking about using non-SNPs instrument as instrumental variables, such as, let's say, polygenic risk scores.
Yeah.
Or with necessary thoughts behind it. Polygenic risk score or SVs, CNVs.
Yeah.
Is there any possibility of including non-SNPs as IVs?
So I think you can definitely include non-SNPs as IVs, but I think in terms of polygenic risk scores, you've just got to. This is what you get with polygenic risk scores. What we've done here is the polygenic risk score for CRP, and you see that the polygenic risk score starts becoming associated with all these different factors. So I think a primary analysis should never be just using a polygenic risk score instrument. You should, at least with the single variants, you can at least do the sensitivity analyses. So you should really think about which of the variants actually are plausible in the gene environment equivalence sense. Yeah, non-SNPs, definitely. In fact, there's a lot, there's a lot of interest in looking at quite low frequency variants, but combining large numbers of low frequency variants. So, for example, there's a variant with allele frequency 0.004 in CRP, which is a complete knockout. It's brilliant. It gives you a large difference, a really huge difference in the phenotype. But then there's even rarer ones, but all in the cis region. So collapsing things which have predicted functionality within regions and combining those into an instrument is definitely doable. Especially with you need big samples for that. But then you get a very large phenotypic effect, and we know from situations like LDL cholesterol that these very large phenotypic effects actually get the right answer about the outcomes. So yes, definitely using non-SNPs is possible, but polygenic risk scores I'm wary about for these reasons in terms of using them as instruments. So, and what say? Yeah, no, so EQTLs can definitely be informative, especially if you take the tissue specificity of the EQTLs. What you want, again, I think what's often done naively is simply wrong. People like, you often see people meta-analyze across tissues from GTEx, for example, and they say the EQTLs that we're going to use are the ones which are seen in all tissues. That's completely the wrong answer. The ones the things which are likely to actually have effects are going to be the situations where they are differently regulated. And there's a paper by Tom Richardson, probably around 2021, in Nature Communications. I'm last on it, which demonstrates that in terms of thyroid EQTLs and one EQTL, which is very differently expressed in thyroid than it is in all the other tissues, and it relates to thyroid outcomes. I mean, it sort of makes sense that the things are going to be where they're going to actually have effects. Is the tissues are going to be differentially regulated where they have effects and where they don't have effects. When you see an EQTL which is general across many tissues, it's obviously it's probably having it's either some sort of cell regulatory type thing which you need to stay alive, or it's just something which is having it's just sort of been allowed to just produce. I mean, there's loads of phenotypes which you just carry on getting it. Insulin-like growth factor 2 doesn't seem to do anything really outside of development, but you get you get high levels of it. In fact, it goes just up to high levels, and you can get genetic variation in it, but it's very unlikely to be doing anything. So no, I mean, I think that phenotypes like EQTLs, MQTLs, methylation QTLs, which are all available through GoDMC, the Genetics of Differential Methylation Consortium. Paper in Nature Genetics, Caroline Relton, who was here earlier this week, led, and we've updated that. It'll be about 6 times larger soon, and then a release soon. So you can get, those are things that are very valuable resources, especially for putting together sort of whole, pictures of possible biological cascades using them.
Sample size that is needed to apply Mendelian, and also while studying disease with like rare incidents like neurodegeneration, for the example. So what is the ratio of exposure versus outcome? Like, because we have a lot of exposure versus outcome might be a little lesser.
So so there's no the issue about sample size is that it depends on the size of the genetic effects on the phenotype. If you have a large effect on your exposure, and it's a common variant, then good answers have come out of samples of a few thousand cases. And with rare diseases with huge effects, it'd be smaller. So there's no sort of like minimum sample size. For those things. I mean, there's a lot of online power calculators which do allow you to put in the genetic exposure effects, the variance in the exposure explained, and get some sort of estimates. But bigger is better, but that's always the case. And for things with large effects, you can see things in relatively small sample sizes. Well, Nidhi has my email address. I'm very happy to answer questions by email. And thank you for putting up with me for 1 hour, nearly 2 hours. That's shocking. Luckily, Caroline isn't here to tell me off for having gone on too long.
Thank you for
No, it's a
wonderful
pleasure.
talk,
No,
Professor George.
thank you.
So different